Hello, everyone, and thank you so much for joining us today. We have a special guest, Steve Pondell, and he's going to be talking to us or with us about INDs, NDAs, and DMFs. If you have any questions throughout the webinar, feel free to drop it in the Q&A. We'll try to get to them at the end. We've got a lot to cover, though. It's going to be an exciting conversation about regulatory, which is one of my favorite topics. So, Steve, can you tell us a little bit about yourself?
Yes, my name is Steve Pondell. I work for a consulting company called IBT Solutions, and we've been working in the consulting area for about 18 years. We work with clients that are usually small to medium-sized biotech companies in the development of their CMC process. CMC is chemistry, manufacturing, and controls, which is making the drug, testing the drug, and putting the appropriate filings together that describe the drug. So those are the things that we provide to clients. I have worked in various small biotech companies for the last 25 years. Before that, I worked for a contract manufacturing supplier that made APIs, active pharmaceutical ingredients. And I cut my teeth for 20 years at Abbott Laboratories in Chicago, doing engineering, manufacturing, quality assurance, not regulatory, surprisingly. I'm an engineer by education, and I do have my MBA. So look forward to the discussion.
Awesome, thanks so much. And Steve and I have worked together in a couple iterations where we've had some good times, good conversations about analytical development and product development. So just to start, Steve, can you tell us a little bit about what is an NDA, what is an IND, what is a DMF?
Okay. An IND is an investigational new drug, and it's actually an FDA document. It's an NDA application. It's a document that you put together when you are ready to go into human clinical studies. So it summarizes all of the information that you have that you want to present to the FDA from a clinical point, from a manufacturing point, and from a non-clinical point, which is all of your animal studies that you've done up to this point. So compiling that all to the FDA, telling them, I want to go into humans, here's my basis, here's all the documents of why I think it's appropriate at this time. Excellent. So then you go through your multi-year clinical process, and you get to the end of that process, and you then compile a new drug application, which is much the same information, but much more of it, to go to the FDA and talk about the fact that you are now ready to take your drug into commercial distribution. So you know how to make it. You've tested it on humans in various protocols. You've shown that it's efficacious, that it works, that it's safe, that you understand it, you can make it. And you put all this information into the FDA, and they agree with you or not. NDAs are for small molecule drugs. Typically things like aspirin and acetaminophen are small molecule drugs. It's a very defined chemical entity. Its equivalent is a BLA, a biologics license application, which is for biologic products, something that you might ferment, an extraction that you might get out of a plant. It's a complex multi-component system that it's very, very similar, but there are some key differences in that. A DMF is a drug master file, and there are multiple kinds of DMFs, but we're going to talk about the DMFs that apply to products that like probably you're all developing. So a DMF is something that you would use, and I'll give the classic example. If you want to manufacture a generic drug, you want to manufacture the active pharmaceutical ingredient for a generic drug. I'm trying to think of a good example for that, but ibuprofen is probably one. You've got Advil as the name out there, but everybody's making some version of Advil off. That's not Advil, it's a generic. So if you are making a generic API, again, active pharmaceutical ingredient, you might sell that to 5, 10, 15 people that will formulate it, put it into tablets, put it into bottles, and sell it. So you have your own filing that says how you're making the product, all the controls that you have on the product, and all your customers have to do is refer to that DMF. The FDA has the DMF. They can go look at it and say, oh, this is what they're talking about. But it keeps your information, it can keep your information confidential. As important, you only have one filing for all of these customers instead of 15 filings for all of these customers. So I think that's the key differences between those three entities.
And what are the regulations and guidances that you refer to mostly when you're working on an IND? And let's maybe just start with INDs.
There's two sources. Many times they overlap. There is an organization called International Conference on Harmonization. It's a global organization that's been in existence for 30 plus years. Prior to the 1990s, every country had their own system of approving products to go into the clinic or commercially. So if you wanted to take your product worldwide, you had to create 25 different packages with the same information presented in different ways. So the idea here was to streamline. There is a whole series of documents regarding quality, safety, clinical studies that basically all of these countries have worked for many years to get a harmonized document. So there's one that's particularly pertinent to us is ICH Q7. They all have numbers. Everything has numbers. And Q7 talks about the manufacture of active pharmaceutical products. And it's kind of a set of guidelines that if you follow the guidelines, then you're pretty much going to be okay. If you don't follow the guidelines, you need to have a good reason why you're not following the guidelines and justify it. The other set of guidance documents is FDA's own guidance for industry, GFI. And there are probably hundreds of them. Every time I look, I find more. There's a guidance for everything. Where they overlap is all of the ICH guidances have also been incorporated into FDA guidance. So the exact same document is in the FDA guidance. So those are the two things I'm looking for. And truly one of the things that we kind of bring, Wendi and myself, is not only knowing the guidances, but knowing that there is a guidance and to go look for it. So know which one to pick up. And that's a skill in itself.
Not nothing. Yeah. And I think also understanding how to apply it. Right. Yeah. And how it's applied. I think that there's specific guidances for DMF. There's specific guidances for INDs and all of that. Maybe now's a good time too to touch on CTD a little bit and how that kind of flows, because there is an ICH for CTD and FDA guidance for
CTD. One of the guidances, as the agencies have tried to make things uniform, harmonized, is presenting the information in that same uniform way. So if things all work out well, if you're writing a section about the specifications for your product, that happens to be one of the sections, it will be in the same place in the document. It will be presented the same way. So hopefully, if you get lucky, you will just be able to cut and paste from one filing to the other. So CTD is the common technical document. And it is the structure, it's the skeleton of how you present your information in filings. It's very, very similar between an IND early in the process and an NDA or BLA later in the process. It's the same structure.
The
There's
DMF.
real difference in how much details. DMF actually does use the same format. If you're making it, you might not use all of the structure because a lot of it doesn't apply, but the part that you do use, it's just a lot easier for everybody, and actually FDA requires it. We also have something, I don't know, do you want to talk about submissions right now, Wendy?
Yeah, go ahead. Yeah.
On submissions?
Yeah. Yeah. So
Okay.
the submissions, kind of what he's alluding to is there's portals that you have to submit these through to the FDA, and it's a little bit more challenging. So do you want to talk a little bit about how we do that?
We can do that. So CTD is the common technical document. eCTD is electronic common technical document. And FDA now requires that. You must submit through the portal. You can no longer, except in very unique circumstances, can you submit a paper copy to the FDA. So it goes into a portal. It has a particular structure, the software of how it goes in. There's a couple things that it does for FDA, and it does for you too if you want to use it. The FDA, when you're talking about your clinical study, for instance, in your IND, you may reference a non-clinical, some safety data. And you can actually do that in the document. You bookmark it and hyperlink it. So the FDA is looking at it and they say, oh, I want to see this safety study. They can click on the link. It takes them to the safety study, takes them back. They can see all of these different modules and move back and forth.
Mm.
But even more so than that is as you update these documents as you get new information. So, I might update the specifications and I only put in the new specification module. But when FDA looks at it, they will see the new specification module along with all the other modules. So they will always have the entire most current set of submissions. So there is a special software for this. I'm sure any of us could figure it out, but part of it is it's somewhat expensive, and you really have to know how to do it. If you don't do it right, you'll get bounced out of the portal, and you'll have to fix it. So it's not a major expense, but there are people that publish, call it publishing, putting these modules into the structure, into the backbone, and then submitting it to FDA. Well worth finding the people. Some people, I think I've never heard of an issue with improper submissions. Some people take longer than others and charge you more than others, but there's a lot of people that can do it. I looked at it for myself and I said, this is not worth the time and effort to do it, specialize in it. It's not something you normally pick up on the side.
Yeah, and it's good to find an expert in it that has experience to have it go really smoothly. So let's get into some detail on the IND. So we talked a little bit. Do you want to kind of refresh just real quick on the purpose of the IND?
Okay. So the IND is the compilation of all your data to submit it to the FDA, and then the FDA will review it and say they agree. This is the protocol you should. We agree with your protocol. We agree that it's safe. We agree that your product is okay, and they will give you a clearance within 30 days. They will tell you yes or no.
Yeah. Mm-hmm. And the clearance is for go forward with your clinical study, right?
Go forward with the clinical study. Yeah. And you can, if you've got one product and one indication, that IND will stay with you all the way through development. So when you get done with the first clinical study, you'll analyze your data, you'll put together a clinical study report, and you will go back to the FDA with the next protocol, which they will review again, and you will update your manufacturing based on what you've learned from the first time. But this is a continue evolving document. Technically, the IND ends when your investigation, when your clinical studies end, and you will convert it into an NDA because all of that same information is there and more.
So what kind of information is the FDA looking for to be able to give approval on the clinical study?
So there's three main areas. And let me say that the FDA at the beginning is interested in, is it safe? Because this is your first time in humans. Like, yes, you will have animal studies, and they'll say, I know what my toxicity is. I know what my efficacy is. I know how well it works in rats, mice, whatever your animal species are. But nothing like taking it into man the first time and finding out if all your correlations are correct. Mm-hmm. Obviously it goes without saying, there's nothing, you can only simulate what happens in the human body so much in other species, and every species has some deficiency that doesn't mimic the human body. So that's why you use multiple species. So you're putting together all of these animal studies. Probably the biggest ones are the toxicology studies, which are run under their own set of regulations and take a lot of time and they take a lot of money, and they're well documented in terms of what the FDA requires. So that's the biggest part of safety. On the protocol, they want to know that most times you're in a phase 1 study, you may be on healthy humans. You're trying to find out how well your drug is absorbed into the system, how well it's excreted out of the system. You need to make sure that you need to justify the starting dose so that you are not at risk of harming the patient because you give them too much before you know what's happening. So, I mean, typically people are ramping up the dose. They start at a very low level and they increase until they get an effect or get a toxicity. They get an adverse event. So the FDA is looking at that study. Are you adequately controlling the starting dose? Who are your patients, or who are your subjects? If you're studying, I don't have a good specific example, but you want to make sure that if you're studying a particular drug for alcoholism, that you don't bring people in that have alcohol abuse problems, or you do. But I mean, you specify who is allowed in, who is not allowed in, and it's a long list. Will you allow pregnant women to be in your study? You probably at that point don't know if it's a problem. So they just say, no, we don't. So that's a very important. The inclusion and exclusion criteria of who's allowed and who's not is very important. The last part is manufacturing. The FDA wants to know that you have a sufficiently characterized process and product that and you know what's going into the humans. Okay, you don't know everything about it, but you are expected to have manufactured the product in a GMP environment, and we can talk about that in a bit, with GMP manufacturers. And the expectation is that you have actually manufactured the product that will be used in the clinical study. So it's not like I think I know how to make it, or I made it in the laboratory 3 times. No, you actually have to have made the product that is going to go into the clinic. And you need to have analyzed it appropriately through GMP laboratories with verified or qualified methods. They may not be fully characterized, but they need to be sufficient to say, I can get an accurate reading of what I'm looking for.
And to clarify, this is for a phase 1 IND.
This is for the very beginning.
The very beginning of phase 1 IND, qualified methods, and GMP manufacturing.
And you need to put that clinical lot on stability. So you need to be making sure that it will retain its characteristics over time. And over time is what you define it to be. Maybe it lasts a month, maybe it lasts 2 years, but the minimum data that you have to submit is 1 month of data. So, I mean, I'm constantly coming into different clients in this pre-IND phase, and they're saying, we're ready to go. We've made product in the lab and we've tested it here and we know what we're doing. And I look at it and say, well, you haven't made it the way you need to make it. You haven't put it on stability. You don't have appropriate methods. So usually one of the first things I get to do is say, you don't have enough money and you don't have enough time in the schedule. So let's talk. That's the requirement. And I've also seen where people didn't manufacture their product ahead of time, or didn't put it on stability, and FDA said, no, bring it. This is what it is. We told you that. And if FDA rejects an IND, you go on something called clinical hold, which means you can't start your clinical study, of course. And they give you a list of reasons, says these are all the things that you need to do. So if your methods aren't qualified sufficiently, they could put you on clinical hold. You need to go out, get your methods qualified, test your product, and then resubmit the IND with another 30-day review clock. So going on clinical hold, especially if you're a company that has to announce that publicly, is not good to start with. You thought you were ready to go, and the FDA said no. It's like, well, do you actually know what you're doing? And you've probably got a 2 to 3 month time hit because you didn't do it right the first time. So it's important to do the best job you can. And I know it's a difficult time for companies. They don't have a ton of money, but this is what you got to do to get in the game.
And they're just the basic requirements, essentially the check boxes that the FDA has to be able to allow a clinical study to go forward. And it's a safeguard to make sure that you're prepared, you're adequately prepared to actually dose humans, right? One thing that I wanted to bring up, you said that the methods only need to be qualified, yet they're testing stability samples, right? And so the stability guidance has requirements that the methods are also fit for use. There's some carve outs for phase specific, but you need to make sure that it's a stability indicating method. Even if it's not fully validated,
Right.
you still need to make sure it's stability indicating.
Yes. And I mean, I suppose you could not do the whole gamut there. You need to show that at least, yes, if it degrades, that you'll be able to detect it. That's the whole stability indicating method. If there's a change, that you will be able to detect it. If it's not stability indicating, then you won't know that it's changing.
Right,
So that
right.
is,
So you need to, yeah,
yeah.
need to be able to demonstrate that. So let's talk a little bit about the pre-IND packet and the pre-IND meeting, because I think that's kind of I think it's a really efficient way to get information out of the FDA.
So there is a process. There's process throughout development that you can meet with FDA. Almost never do you get to call them up on the phone and say, I have a question, I want to talk to you. There's a process. You put in an application, a request that we would like to meet with you. And let's talk about pre-IND. The other ones follow similarly. This is voluntary. You do not have to have a pre-IND meeting, but I do encourage people, if they have the time and can, to have the meeting. So this is a chance to ask whatever questions you want of FDA, typically 5 to 10 questions. It's all they want to see. And usually the questions that you're asking, I do ask some standard questions. I always ask about my specifications. And we always ask about the clinical protocol because they're going to have comments on them. So this is a chance to get free advice from FDA before you get rejected because you didn't do it right. So we have a chance, it's typically several months before we actually submit the IND, to have this discussion with the FDA, to go through the questions we have and have their responses to them. Again, we're going through this right now with one client. We just submitted a request. FDA has 60 days to have the meeting. So again, you got to build that in your timeline. We submitted a list of questions with about a paragraph along with each question, justifying or explaining the question. So for instance, we went through the specifications. We said we have a drug substance specification, we have a drug product specification, does the agency agree that these are appropriate for phase 1? And they will come back, and I am fully expecting that they will say yes, but, or do this, and that's great. So the next thing we need to do with this client is within 30 days, we need to submit what's called a briefing package. And that is the background and justification for what we have for our clinical study, our non-clinical, all the things that we've asked questions on. This is all the background. This is the justification. We've done this study to show that it's safe. We asked you if it was okay, and here's the information from the study. Specifications are in that document. We're also asking about our manufacturing process because there's a few things that we're doing that are a little bit outside the guidance, and we're trying to justify that and explain it and get FDA's okay with it. We think we're okay, but they will tell us if they are or not. And we will then get a response back by the end of June, which will say yay, nay, we agree with you, we don't agree with you. And now we've got some time to fix those things before we put the IND in place. It's called the pre-IND meeting. I cannot remember the last time there was actually a meeting. If they can answer it by answering the questions on paper, they will. And most of the questions, I mean, this is where you bring, people like myself in to say, how do you word the questions so that you ask the right question and get the right answer? If you put a general question out there, you're going to get a general answer, and it isn't going to help you at all.
Then when you file the IND, it comes back and then you're on clinical hold. Yeah.
It could, or you might as well not have had the meeting because you didn't really ask a good question.
Right. Yeah. Yeah.
So it is important to do this, and it also, I mean, I don't know the inner workings of the FDA, but they generally have the same people looking at these. It's not like they bring in a new team every time. So this is your first introduction to the reviewers, and when they see the IND, then hopefully it goes better because you've asked intelligent questions and they've given you intelligent answers.
Yeah. So you said that the IND kind of follows the product through the development. So from phase 1 to phase 2, you submit an update to the IND that is for your phase 2 or your phase 3. How do you see the product development progressing from, let's say, your initial IND into phase 2 into phase 3, specifically around the CMC?
Okay. Yeah, and this is where the FDA has a lot of guidance documents. They're not part of ICH. So I talked about GMPs a little bit. That's as good manufacturing practices. It's a set of regulations and guidances that tell you how you should be making your product. They explain, for instance, what does method validation mean? It's there, and it tells you at different points in the process how far you have to be from a manufacturing point. I mentioned that the initial clinical product has got to be made in a GMP facility, which means you have qualified your equipment, your utilities, you have documentation systems to control everything, you have exception systems. If something goes wrong, it tells you what to do. All of those things are in place. What may not be in place for phase 1 is you don't know much about your chemistry. And the FDA knows that, and they don't expect you to have complete control over something that you've maybe only made 2 times. So your limits may be wider, your ranges may be bigger because you don't really know yet what's required.
And
So
on your specifications, you might be just reporting. You might be collecting data and not have limits set yet.
And I will say, by the time you get to the final NDA specification, they don't accept the report. No. Either you have an answer or you don't have it in the specification. The specification is not for information gathering, especially at the later points. It is for controlling the product. If you want to gather information, do it some other way.
Yeah. Yep.
So as you go through, for the IND, you've made at least 1 batch for your phase 1 study. You probably only made 1 batch because it's a very small study. But you are starting to get ready. As you get into phase 2, do we need to touch base on what phase 1, phase 2, phase 3 is?
I think that's probably a good idea. Yeah. Yeah.
I'm going to give a classic example. This can change and you can justify it, but classically, phase 1, as I mentioned, is where you're checking safety and you're checking dose. So typically you'll start at what you have figured is your minimum effective dose. It's like, or even lower. So I put this first one out here. I don't even expect to see a response because it's so low. And if you do, then okay, it's a surprise, and that's okay. But typically you're taking a handful of patients, 3 or 6 patients, putting them on the drug for a period of time, and you're looking to see how much is in the body. You're also looking to see any adverse events. Does the patient have something, especially if it's related to your drug? And you're continuing to increase the dose, typically until you either get to a target concentration. More typically, you're increasing the dose until you start to see adverse events. And then it's like, okay, I don't want to be that high, come down one. That's your end of your phase 1. And many times, again, it's done on healthy patients because you don't want your somebody who has your indication in that study. I want to know what a normal person, a normal healthy person would do. Phase 2, you are into patients, but you're now trying to find out if it works. So now you're looking at patients, very specific subset of patients, and you are looking to see, does it work? You're looking at symptoms, symptom improvements, or not. You're seeing what happens with the drug in patients in a fairly small study. This might be 50 to 100 patients, probably not placebo controlled. So you're just seeing, does your drug seem to work with patients? Phase 3, and usually the requirement is that you have 2 phase 3 studies. And these are the big ones. Depending on your indication, it could be 300 to 3,000 patients. You're probably at multiple clinical sites. This is the one where you've opened up that exclusion and exclusion criteria to take more people. That does it work on your entire population? It's depending on the indication. It's probably controlled by a placebo. So some people are taking a placebo that doesn't do anything, and you're comparing them to people that are taking your drug. And this is all patients. So at the end of this period, with your 2 studies, you're doing a very detailed data analysis, statistical analysis, and out of that, you determine whether your product works or not, and whether you're ready to go forward for commercial, an NDA, and commercial distribution. So each one of those, you want, go ahead.
Yeah, so to kind of bring it back around to the CMC, we talked a little bit about methods and qualified and all of that. Maybe we can talk specifically about going into phase 3, what it looks like, and then post phase 3, what that looks like before you submit your NDA.
Okay. Let's work backwards a little bit. For the NDA, for the IND, you just had to show you knew what you were making and that it fell within a certain set of ranges and specifications. For the NDA, they want you to show that you know how to make it, you know how to make it consistently, you know about your product, you know what the limits are, what's good, what's bad. All of the uncertainty at phase 1 is gone. You need to know what your product is doing. And really, you only get to know what it's doing by making it.
Right. Well, and I think
yeah,
you need to know, and you need to have the data to support it.
Yep,
so
absolutely.
Methods are validated. Stability data supports the shelf life that you're asking for. Process validation, all of those things. Yeah.
Right. So going to the beginning of phase 3, or even before phase 3, I mentioned how many patients you got on the study, which means you need a lot of drug. So you are probably moving to a different manufacturer that's at a bigger scale. You may or may not be moving to a different laboratory to be able to support commercial manufacturing. You're not manufacturing commercial yet, but you need to be getting close to that. So one of the first things, well, 2 things you need to do. You need to do a process development study. This is extensive and pretty expensive. You need to understand your process. You need to justify, again, with data. I need to heat this reaction up to 60 degrees C, and I need to hold it for 10 hours. Okay, so now you're going to go in the laboratory and you're going to challenge that. All right, what if I only hold it to 55 degrees? And what if I only do it for 5 hours? What happens? Where's the edge, or at least where's an operating envelope I can use? So from a process point, you're doing that with everything. Where do your impurities come from? Where do they go? It's important to know all of that. from a method point, you are now getting to full method validation. You are really understanding your methods. You're proving that they can be run by any qualified technician at whatever laboratories you're going to do. So up until now, it might have been one instrument, one technician, and going forward, it's going to be any comparable instrument and any comparable trained technician. So all that's documented. It's in protocols. So before you do the work, you define what you're going to do and what's acceptable. One of the first things you're going to do probably at the beginning of phase 3 is what's called the registration lot. We talked about stability. You're going to go into your NDA and you're going to claim that my product is stable for 2 years. Well, obviously you need data that says it's stable for 2 years, and you can't, just like being pregnant, you can't be pregnant for 4.5 months. You got to go the whole time. So you're making product at the process you plan to use commercially, at something close to the scale you plan to use commercially, and you're putting it on stability. And that is, again, when I come into a company that is late phase, they've usually left registration lots too long, and they're going to have to submit with less data than they wanted to. And again, that is almost always a 1-for-1. If you want 2 years of dating, you need to have 2 years of data. And that goes into the NDA.
Yeah,
The NDA,
yeah.
9 months to review. I mean, it's a long process, but it is early.
And the ICH is really clear. It's ICH-Q1A is really clear on how much stability you need based on your storage conditions. So typically it's 6 months accelerated, 12 months real time. But it could be, full shelf life depending on what your storage conditions are.
Yeah. And you might have a product that's only good for 6 months, and that's fine.
Mm-hmm.
You'll know it, and then you'll know that you've got to turn that over regularly. The next thing that happens from a process, analytical should be ahead of this process, but the process will need to be validated. So you will be making 3 commercial scale batches, at least 3 commercial scale batches, and you will be testing them. You'll be stressing them somewhat during production. This is proving consistency. This is proving you can make the product consistently 3 times, at least 3 times. Very expensive. It's time consuming because typically you've got your API, it has to be validated. And before your formulated product, your drug product has to be validated. This is typically a year to 18-month process. And you need to have this done. So the budgeting and putting the time for the time and money that it takes is a key. Most companies, by the time they get to phase 3, they understand what's coming up, but they usually haven't put enough money in the budget to do what needs to be done.
So I think that's kind of a good time to talk about the gates in product development. It's like a go, no go decisions, right? So, first IND approval by FDA or other regulatory body to go forward with the initial clinical study. Then I would guess it was passing, having a successful phase 1 clinical study, right? And then moving into phase 2. And so do you see your customers then, having kind of phase appropriate budgeting? Like if we pass this gate, then, right?
Nope.
No. Okay.
No, and that's okay. I mean, this is why we come in, is to help them understand that, especially going from phase 1 to phase 2. There's a lot of thought. I've done it. I'm approved, and technically you are. I mean, the FDA is not usually going to expect more in phase 2, but you're going to get caught if you don't learn your process and learn how to scale up. You're going to get caught when you go to phase 3 because you haven't done anything. So then really going to hit the road when you have to find a different manufacturer, when you have to scale this up, when you scale it up and realize it doesn't scale up because there's some parameters you didn't understand. So again, it can be, and you need that product to get your phase 3 studies going. So yeah, it's a challenge, but I also understand it. I mean, if you look at uh, um, small companies and where they are financially, getting through the phase 2 study, the proof of principle study, I mean, that's a key milestone. And until that, you're probably, unless you're lucky, not going to have enough money to do your phase 3 study until you get done with the phase 2 study. So, I mean, yes, it's a natural gating from the investors, but if you had the money, it'd be really nice not to get stuck doing it all at the beginning of phase 3.
Yeah, because if you're waiting for that phase 2 success, I guess, then you've lost some critical time there where you could have been doing some of the work to get you the scale up and everything to get you into the phase 3. Yeah, and then I think the same is true then during the phase 3 trial. Maybe you're holding off on process validation, waiting for the successful phase 3 trial, and right.
If you do that, you are seriously in trouble. Because you need to have that before you put the NDA in. So at the end of phase 3, typical time critical path, you get done with your clinical study. You do data analysis, which probably takes you 6 months. You got to make sure the data is valid. You got to run it through all of your pre-established protocols. But once you get done with that, that should be the last thing before you compile and submit the NDA. So if you have to stop, wait another year for process validation, that's a big deal.
And I think, because your process validation lots go up on stability as well. And so, getting all of that done. So, we have one question that came up. Did I understand correctly that if we have 30 days from the time the FDA responds to pre-IND questions to submit the IND?
I'm sorry for that miscommunication. For the pre-IND, you submit your request with the questions on day 1. On about day 30, you submit the briefing packet so that the FDA now has all the information they need. And 30 days later, they give you the responses or the meeting or however they're going to respond to you. After that, there is no timeline of when you have to have the IND in. You could take 6 months. You could decide you're never going to submit it. Once you submit the IND, the process to actually review the IND is 30 days. So 30 days keeps coming up here, but it's separate 30 days. And there's no connection between the pre-IND meeting and the IND. Like I said, you don't even have to do the pre-IND meeting.
And once you're like, if you get questions or you get put on clinical hold, is there a clock there? Do you have a certain amount of time before you have to resubmit the IND? It can remain open.
Remain open as long as you do. I had a client that got on clinical hold because they had not made their product and had other problems. And they decided they weren't going forward with that formulation and got a new formulation. So they killed the IND, the first one, and got a new IND for a different product, basically.
And that's like getting a new IND is like requesting an IND number from the FDA, right?
Well, that's the first step, and that's a different part.
Yeah. We had another question. If we have a compound we want to submit a DMF for, can you remind me what we need to include in that DMF packet besides ICHQ7 for it to be accepted and ready for IND? So what do you need to have in a DMF packet for a IND, I think is the question.
Let's talk about it just for manufacturing, because that's typically what you would have is just manufacturing. So if you are looking at an API, it's exactly the same information. It's in a CTV format. It's not an IND has non-clinical stuff, clinical stuff, and manufacturing stuff. A DMF typically has just the manufacturing stuff. And depending if you are doing a drug substance and a drug product, that's 2 parts. So you may only have one of the parts. You have the drug substance
for the DMF. Can you clarify when you say manufacturing stuff? What is that?
The manufacturing has its own module. It's module 3. Interestingly enough, some genius somewhere decided that title of that was not manufacturing, but it's quality. I don't know.
And that's the CMC, so it's chemistry and manufacturing, right?
Chemistry and manufacturing.
Yeah.
So there's sections in there that describe the molecule. So let's talk about small molecules easier. That describe the molecule. It's, here's the structure, here's the weight, here's the solubility, whatever is appropriate. You'll have a section in there who makes it. So the GMP manufacturers are listed. You'll talk about the process. How do you make it? And this applies to drug substance and drug products. I won't repeat it all. Talk about impurities that specifications and test methods. And an important part is justification of test method of specifications. Okay, I've got I got a purity range of 80 to 120%. Well, industry standard and the expectation is 90 to 110. So tell me why you can't hit 90 to 110. That's where that goes. You'll have a section that talks about the primary packaging. What are you putting your drug substance or drug product into? And is that an appropriate thing? Is plastic okay? Is clear glass okay? The answer is yes or no. And then your stability data is in its own section. So each of those is a subsection, and each of those subsections has subsections. There's quite an extensive backbone there, but again, that's the information. Drug product, it's the same. It's actually the same structure with a different letter on it.
And I think the best advice there is to look at the CTD backbone, and in the ICH, and I feel like it's an M, like M5 or, yeah, I can't remember. We can look it up, but look at that, and there's actually guidances that tell you exactly what needs to go. So it's 32S1 is description or characterization, or then and then you have structural elucidation. It tells you exactly, and it tells you exactly what you need to put in. There's a specification section, and then a subsection that's a justification, a method section, and there are validation sections and reference standard sections, and it really there's a lot of really good information out there on exactly what you need to have in that. Yeah.
Yes, there is. And I will say most of that, just as a caveat, that's mostly tied to NDA. It's the same information in an IND,
right.
but many times it's not as extensive. And there's other guidances that say you don't have to do that, or you don't need as much, but yes.
You don't need certain sections for the IND. Yeah. And that would be one of the guidances for industry from the FDA, not necessarily in the ICH. Yeah.
That's an FDA.
Yeah.
There is a guidance there that is very descriptive of what goes in each section. So
yeah. And it puts people like me who love to organize things in a very happy place drafting those. I think it's great. We had another question that says it's a question about the process overall. So the question is it DMF, then IND and NDA? Do you want to take that? So the question is, what am I understanding correctly that the process overall would go DMF, then IND, then NDA? Do you want to talk about how the DMF fits into the IND to NDA process?
Yeah, and I will say first, it really doesn't.
Right.
You do not need a DMF. Most innovators don't have a DMF because it's your product. You don't want anybody else to make it. So
I think we could say that in that case, then it would be vertically integrated. So you would be manufacturing both your API and your drug product typically. And the purpose of a DMF is when somebody else needs to refer to your application for their application. So it's kind of a separate entity from the process.
Well, and most times, and the example I use, most times DMFs are used for commercial products because most times you're not taking your clinical product to more than one place. But we have a client that is they're doing clinical development a little differently right now. They're doing all of their development through investigator-sponsored studies. So some researcher at different institutions or hospitals has an indication they want to study, and they want to use our client's product. And there's multiple indications and multiple centers, and they don't need to deal with each other. They each have their own study, but our client is providing them with the same product. So rather than have each of these investigators at these different hospitals creating their own CMC section based on my client's product, and then having to control that, a client has just said, we have a DMF, you can reference that. And we will control the DMF, and we will make sure the DMF is appropriate with the FDA. And you don't have to do that. And that has worked pretty good. The challenge that we have with that is that the FDA is not used to having DMFs in clinical studies. They're used to seeing them with commercial products. So Wendy and I have spent a lot of time together trying to get test methods for this client up to commercial standards, because that's what the FDA has been requiring, even though they're in a clinical study. So their methods are pretty darn solid right now because the FDA has continued to push them to get there. It's probably a good strategy, but we never thought about it ahead of time of what it was going to mean.
How much work was going to go into getting that DMF there. In my experience, because I did a lot of work in generics, so all of the DMFs we were looking at were DMFs for either an API, or so like our vendor would have a DMF, and we would then get a letter of authorization from them to be able to refer to that DMF in our application. So typically that was an ANDA, right, which is the abbreviated new drug application for generics. But DMFs are also used for critical excipients, like if you've got an excipient that controls release.
Could be.
We've had DMFs for that as well. So DMF really is useful when you're using somebody else's material, when you need to refer to it, when they don't want to give you all the information. I've also seen it from contract labs. They have a DMF where their methods reside. They can upload their methods to that, and then their customers can refer to that DMF, so they don't have to give their IP away and their method validations away to be included in the application. It simplifies its change control.
Yeah, sometimes it's confidentiality. I mean, most times in NDAs or DMFs or INDs, the commodities, the vials, the plastic bottles, we buy them from somebody and they have a DMF, and we just say we're buying this. That's the DMF, and it's also simpler for us. We don't have to put all that information in our filing. We just refer to their filing.
And I think in that example then too, if they had their information in 15 applications and FDA comes back with a change in response to one of the applications, it could have a ripple effect throughout all of the other applications and require then submissions by all the rest of their customers. And so then the DMF keeps that really simplified there. So
it simplifies it, but I mean, one of the things that with this client that we have to do is every time we make a change, we have to tell the clients that we made a change. We generally tell them what it is, but yes, we have to notify everybody that is authorized to refer to the
change has happened. Yeah,
just happened.
So I want to move in. We've only got 5 minutes left, so I want to pick your brain on maybe advice that you can give to the folks that are listening to this. So what are some of the most common mistakes you've seen in an IND or an NDA or a DMF?
Well, part of that is not understanding your impurities. I mean, impurities are very big. You typically will run, I mentioned at the beginning, you will run these toxicology studies under very controlled conditions. And you're not only checking the tox of your compound, you're checking the tox of the impurities. So if you don't know where your impurities come from and where they go and how you control them, you could get into manufacturing at phase 2 and find an impurity that you didn't know was there and wasn't covered in your tox studies, which means you got to go back and do another tox study on this impurity. So that's very expensive, very
Yeah, we talked a
expensive.
lot about that in the impurities and in the specification. I think, and in the analytical development webinars about how important it is to do that impurity assessment early and make sure your methods are specific so you can quantify those as early as possible, or it does get very expensive. Yeah, yeah.
Absolutely. And again, the same client, we had 2 different instances where we found a new impurity. We had a peak in there that we didn't know what it was and had to go figure it out and
Mhm.
justify it. Yeah, it was a very extensive and expensive process.
Yeah, yeah. Where should companies not cut corners?
I guess I would say not cutting corners would be in the phase 3 part in particular. If you can, sometimes the FDA will grant you some leeway going into phase 1. If you've already made product somewhere, it's a big process, and and you didn't make it under GMP manufacturing. Um, you can probably put a justification there. You can tell them why. Here's the gaps. Here's why we think it's okay. You won't be able to do that for the NDA. I mean, they'll bounce it out. Um, so that's the later you go, the less leeway you have and
Mhm.
should have. But I mean, that's part of where bringing people like us in before your IND is we can kind of tell you. Most companies at this point are saying, I have all of these things and I'm ready to go. And we take a look at it and say, well, you're not really, but maybe there's some things that we could defer. We could put a band-aid on for right now and justify it to the FDA. And that's what you
Mhm.
hire us for, is to put a smart justification. Either you can do it, or we'll say, you're going to get rejected if you try to do that. So I think
Yeah.
there's places to take risk early, but it needs to be well thought out and well justified.
And I think too, the understanding the everything that's going to need to happen. So there's a clear understanding of the time and the cost associated with drug development from IND to NDA.
Yeah, and again, we talk about phase 3, but when you get done with phase 3, your whole organization is going to ramp up. You're going to need more people. You're going to be making more product, more batches, and most companies at phase 2 are putting the blinders on. They might know it, they don't want to talk about it, but
Quality
they need
systems, the whole everything. Yeah,
everything
yeah,
to change.
yeah. So last question, what is the one takeaway you want people to remember?
There's a lot of information out there, a lot of requirements and guidances that need to happen along the way. And I would just encourage you to make sure you're asking the right questions early enough of the right people, because it's not like we we're we are gurus because we've been doing this so long and we have the scars to show when we've done it wrong. So whether it's us, somebody like us, somebody that you find that you hire, it's like ask the right questions early so you can adapt. If you haven't done it before, there's a lot of things you're going to find out, and they're
Yeah,
all going to cost me.
yeah, they're all going to cost money. It's not cheap. It's not
It is
cheap,
not
and
cheap.
I think it's also not for the faint of heart. Right? There's a lot of anxiety and risk that goes into drug development. So yeah, excellent. Well, Steve, thank you so much for taking the time to do this with us. I think that I hope that everybody learned a lot. I think it was really informative. I'm going to put a slide up here with our contact information. Let's see here. So if you've got any questions, you can contact me at engage@triveritylabs.com or contact Steve at spondell@ibtsolutions.us. Steve, thank you again so much. And we'll see everybody again in a couple of weeks. We're going to be talking about ICH Q14, which is analytical development, and probably a little bit in ICHQ 12 as well. Thank you very much. Thanks, Steve.
Thank you for having me.